Hello everyone, welcome to today's webinar, which is organised by the journal Biofabrication. My name's Richard Calsall and I'm the publisher of the journal Biofabrication. Today's webinar will run for 60 minutes and we will finish at 4 p.m. GMT. Before we begin the webinar, 2 points on housekeeping. Firstly, questions from the audience may be put forward at any point using the question section on the webinar panel. So please do make use of this. There'll be a 15-minute question and answer section at the end of the webinar, and we will endeavour to get through as many questions as possible. And as a polite reminder to the speaker, please do ensure your camera and microphone are both enabled during the question and answers section. So the webinar will be structured as follows. I will begin with some very brief words about our organisation and the journal. And then without much delay, we'll move on to the main event, which is a scientific talk from Dr. Shrike Zhang. And as indicated, the webinar will wrap up with a 15-minute Q&A section at the end. So today's webinar is organised by the journal Biofabrication. Biofabrication is owned by the learned society publisher, the Institute of Physics Publishing. We are a charitable organisation and have over 70 journals in our portfolio, ranging from topics covering applied physics through to environmental science, material science, biomedical engineering, and medical physics, and so on. Biofabrication is IOP Publishing's top-tier biomaterials journal, and Biofabrication specifically covers work reporting advances in the automated processing of biological materials together with other materials to engineer complex therapeutic products such as 3D fabricated scaffolds to assist with tissue engineering, and also engineered biological systems such as in vitro disease models to assist with toxicological screening of drugs and furthering our understanding of disease progression and so on. So please do visit the journal scope page for a full overview of the scope. We are proud to be the official journal for the International Society for Biofabrication. Our impact factor is 9.9. The peer review times on biofabrication are very competitive. We have a submission to acceptance time of 113 days as a median, and that includes time taken for authors to make their corrections. We are listed on all major indexing services, including PubMed Central and Medline. Finally, we are on Twitter, so please do give us a follow using the handle @biofabrication if you're not following us already. And please keep us in mind for your next paper. So without further ado, I'll now move on to the main event, which is a talk from Dr. Shrike Zhang. Shrike is an assistant professor in the Department of Medicine at Harvard Medical School. He's also an associate bioengineer working within the Division of Engineering in Medicine at Brigham and Women's Hospital. Shrike's main area of expertise includes bioprinting technologies, development of organ-on-a-chip technologies, and tools for bioanalysis. Shrike is the author of over 270 peer-reviewed articles and was recently included in Clarivate Analytics' list of most highly cited researchers. Shrike is a member of this journal's editorial board and also holds positions on the editorial board of several other well-established journals. The title of Shrike's talk today is 3D bioprinting utilized towards human disease modeling. So I shall now hand over to Shrike for him to begin his presentation. Thank you.
All right, thank you, Richard, for the invitation and also for the introduction. So hopefully the slides are showing up right now. Yeah, so today I'm going to talk about some of the work that we have been doing over the past few years, primarily regarding how we have been using bioprinting for disease modeling applications. All right, so a little background to start with. So we have been working primarily on two different areas. So first one is human disease modeling, which is today's topic. So rationale for that is, as we know, the conventionally used models either based on two-dimensional static cell cultures or these animal models, they don't really translate in human system because they have the vast differences from human body. So that's why in the past 10, 15 years, people have been really trying to develop these so-called organ chip devices or high content tissue models to allow modeling of these human tissues and organs in vitro in a better way, right? So then you can imagine you can use these models for really improving our capacity in drug development. And also if you combine these models with cells that are coming from different patients or human donors, then you can start personalized medicine by screening drugs or therapeutics according to different people's characteristics. So, but then you can also imagine all these different technologies that are used for tissue model engineering can also be applied to the conventional kind of tissue engineering applications, basic generation of this functional human tissues and organs for regeneration. But these are more longer term goals because everything that you have, you make here has to be basic 100% functional before you put them back in human system, right? So then that's why this is becoming a longer term goal. But in the case of tissue model engineering, a lot of times you don't have to make them 100% functional, but 80-90% functional is usually sufficient for the purpose. So with all those in mind, we have been really trying to engineer various kind of technologies, including biomaterials and all the way to biofabrication, primarily bioprinting, and down to like biomedical devices like this microfluidic chips devices and also the bioanalysis. One of the interesting things I've been trying to do is the combination of, for example, biofabrication like bioprinting with on-chip devices to allow really high content modeling of this human disease, like human tissue and disease models for different applications. So today I'm going to primarily talk about bioprinting for this application. So as we know, human organs are pretty complex, and their microscale tissue field units are also complex, having multiple kind of cell types coming together and the multiple ECM types coming together in the right architecture in order to for the microscale tissue to be functional. So the challenge is here really how to mimic the complexity of native tissue microarchitecture and how to reproduce the synergistic interactions of the multiple cell types. So then we have been, well, we know that there have been many different biofabrication or kind of fabrication methods in the past decades trying to engineer tissues for various applications. For example, use of porous scaffolds, where for tissue kind of engineering, tissue model engineering, you can kind of knit fibers together into three dimensions for that purpose. You can also use some of the like self-assembly methods to allow that to happen. But then a lot of these methods, I mean, they have been really great for modeling tissues or engineering tissues. But then issues with those are, I mean, they're mostly based on manual operations, meaning that if you make one copy of the tissue, that's probably okay. But if you make tens or hundreds of copies of tissue, that can be pretty labor intensive, and also there's reproducibility issue, right? So that's why in the past 10-ish years, people have been really trying to focus on this bioprinting method because it's truly automation kind of a strategy, allowing you to really pattern the cells and ECMs in 3 dimensions with pretty nice control over their architectures, right? So, and also with automation, you can start to really fabricate the tissues at larger scales. So within that whole bioprinting field, we're pretty much interested in every single aspect, from hardware self-engineering, basically building our own printers, to biomaterials engineering, bioinks, and all the way to downstream applications, including what I'm going to talk about today, disease modeling. Here are some examples. For example, I've been really into multi-material bioprinting because, again, as I mentioned, the capacity to building these multi-material kind of ability into the bioprinting is really becoming very important to model tissues, and we have been really into how to design multi-material bioprinting methods for allowing that to happen. And then we have been quite extensively working on this Canon tissue bioprinting, basically bioprinting of hollow tissue microarchitectures that are widely present in the human system, right? So the vasculature and all the other hollow tissues. We have also been working extensively on bioink designs. Again, bioinks are perhaps one of the most important components of bioprinting because they not only have to allow bioprinting to happen in the first place, but also after bioprinting, they have to secure that the bioprinted tissues are viable and functional during the course of culture. So that's one of the kind of most important components of bioprinting, per se. So another area that we have been quite heavily working on recently was minimally invasive bioprinting, basically how you can kind of bring the scale of the conventional desktop bulky kind of bioprinting systems into a smaller scale, and then combine those with, for example, handheld devices or these casted devices for allowing situ mini-invasive kind of in vivo bioprinting or delivery. So today I'm going to more focus on some of these different examples in these different categories and talk about how this have been really used for modeling of human diseases for different applications. So one of the first examples I'm going to give is multi-material bioprinting. So this is actually some work that we have been we actually worked on back a few years ago that we termed rapid multi-material bioprinting. So what happened here is that we combined outlets of the different bioinks into a single nozzle, which we term as a multi-channel but single nozzle kind of bioprinting or extrusion bioprinting system. So why that important, right? So if you look at videos on the right, for our configuration, because everything is coming out from a single nozzle, you don't have to physically switch nozzle. We try to change different bioink, right? In the conventional multi-nozzle format, you have to really physically change a nozzle when you're trying to change a different bioink. So time that's needed for changing this nozzle can be as much as the bioprinting process itself. So that's potentially time consuming, especially if we're trying to have multiple materials come into place. But in our case, because everything again is coming from the same nozzle, then it's pretty fast and much faster than the conventional multi-nozzle based platform. So some applications of similar kind of configurations, one of the applications into this modeling is, again, back a couple years ago, we have been working with some colleagues in Netherlands, really trying to use multi-material bioprinting, extrusion bioprinting to allow verification of these brain tumor models. So what happened here is that you had first printed a kind of little mini brain model that we call, and then that's basically having the macrophages inside. And then into this mini brain model, you can actually print another little volume of this like a brain tumor kind of volume. So that's allowing you to really allow the interactions of the brain tumor cells and surrounding macrophages or immune cells in this three-dimensional biomimetic microenvironment, right? So looking from these graphs, you can see that cells, whether they're being macrophages or these surrounding, all this brain tumor cells, they were behaving much better than when they were cultured on two-dimensional monolayer kind of format. So I'm not going to go into details here, but then the message here is that you can really use bioprinting to allow really flexible configuration of different sort of formats, right? So single cell culture or multiple cell culture together, and then to really allow investigations of all these interactions in 3 dimensions. So then you can basically print multiple copies of these mini brain models to allow, for example, screening of therapeutics that are either based on chemotherapeutics or immunosuppressive therapeutics, right? So then that's one of the applications, and we have also been working on some other iterations where we have now been able to use the similar models to screen, for example, stem cell therapies on brain tumor cells as well. That's coming up hopefully soon in another publication. Then going to a little bit of the cannon tissue bioprinting. So as we know, a lot of the hollow tissues are present in human system, right? For example, vasculature, which is, again, pretty much everywhere in every type of tissue that we have in the human body. So here's the sacrificial bioprinting that people probably know pretty well. So what happened here is that you have a bioprinting system that's allowing printing of these sacrificial microfibers in the first place. And then surrounding these microfibers, you can either cast or print another hydrogel matrix. And once you have cross-link that hydrogel matrix, you can selectively remove this microfiber from the matrix, allowing creation of this hollow perfusible microfiber or microchannel that's emulating the actual fusible vascular kind of microchannel, right? So then here are some examples of showing that perfusion to happen. So this movie doesn't play seams, but then you can actually see the perfusion of the channel if you do the perfusion. But then since it's biologically relevant, like hydrogel structure, you can also endothelialize these microchannels to make them biologically functional, right? So endothelial cells were growing pretty well and became confluent after a few days of culture. And you can also do the branching structure with a sacrificial bioprinting method. All right, so then not only can you create a model of the healthy vasculature, but also you can create disease models of the vasculature, for example, deep vein thrombosis, right? So once you have printed these microchannels and then endothelialize these microchannels, you can then start to infuse these microchannels with, for example, human whole blood, and then clot the blood into the channels. And then you can actually create very simple but biomimetic model of the human thrombus. And if you do histology at the cross section on the left is our model, right? As you can see, actually, very nice lining of the endothelial cells on the interface, and then matrix on one side, and then the aggregated red blood cells on in the lumen area. And if you compare this histology with the histology of a real human thrombus, you can actually see that they are very much similar to each other, right? So again, having very similar structures, having the matrix on one side and endothelial cells on the interface, and then red blood cells in the middle of the lumen area. So meaning that you can actually use a sacrificial bioprinting to allow really convenient fabrication of a human thrombus model in vitro for a lot of the different applications, for example, fundamental biological studies or thrombolytic agent kind of screening as well. So then another thing that not only can you have the matrix in the surrounding that's only having a gel, but you can also embed some other somatic cells into the surrounding matrix, right? So for example, here we actually created a vascularized liver cancer model by encapsulating liver cancer cells in the surrounding matrix. So now you're basically having the channel that's endothelialized, and then the surrounding matrix having the liver cancer cells. So then if you apply drug molecules by perfusion into this endothelialized vascular channel, you can not only see how the drug molecule is actually breaching the endothelial barrier, exerting toxicity over there, but also how the same drug molecule is starting to exert their toxicity in the surrounding liver cells in a time dose, and more importantly, a diffusion dependent manner. This is actually very important because this is very biomimetic, because if you take a drug in like with your either like with mouse or with IV, then the drugs are most likely carried through different tissues with vasculature, and depending on how far away the tissue cells are from the vasculature, there's going to be diffusion dependent effect of toxicity or efficacy that can only be recapitulated in a model like this, three-dimensional, not necessarily bioprinted, but the three-dimensional biofabricated model with the microchannel inside, right? So because if you do the conventional two-dimensional static cell culture, if you apply drug molecule in there, then all the cells are going to be experiencing the same dose at the same time, which is totally not physiologically relevant, right? So this is basically showing the importance of having this more sophisticated disease models for drug testing applications. So then you can also, of course, very meticulously tune the matrix properties of the surrounding kind of construct, and then allow controlling of the permeability profiles or mechanical profiles of all these kind of surrounding matrix to allow or to further tune the diffusion profiles of the drug molecule. So another interesting thing that we have been observing is that if you, again, as mentioned, the channels can be endothelialized, right? So if you culture these channels for long enough, you can actually start to induce a secondary sprouting of these endothelial cells into the surrounding matrix, forming basically the multi-scale vasculature, right? So these are actually lymphatic endothelial cells, basically forming a multi-scale lymphatic vessel into the surrounding matrix. So then, again, you can do this modeling with this configuration, right? So you can actually culture lymphatic vessels on the microchannel surface, and then in the surrounding matrix, you can embed these breast cancer cells, and then you can look at how the lymphatic cells are basically sprouting out into the surrounding matrix and interact with the surrounding breast cancer cells in a VEGF dose-dependent manner. So that's basically allowing you to really look at how lymphatic vessels are interacting with surrounding like cancer tissues in a lot of disease conditions. So you can also reverse the configuration, right? So here we actually modeled a more localized breast cancer kind of configuration, basically mammary ductal carcinoma that are breast cancer cells that are originating from these mammary ducts. So once you have created these sacrificially bioprinted microchannels, you can, now you don't have to endothelialize them, but you can actually seed breast cancer cells into these microchannels. And then with culture time, you can actually see how the breast cancer cells were growing into confluency, and when they became confluent, how they will also start to invade into surrounding matrix, basically showing this invasion kind of phenotype. This confocal image showing the same process, but at high resolution. So not only can you see how the breast cancer cells are invading the surrounding matrix, but also how they were actually having this inward aggregation, which is actually something very similar to the in vivo configuration, not only having invasion, but also the kind of inward aggregation within the memory ducts. And if you look at their basement membrane expression, for example, collagen type 4, you can also see their nice basement membrane production by these cancer cells. And then if you look at EMT transitions, looks like these invading cells were having more expressions of these encadhering than ecadhering, meaning that the cells that are going into surrounding matrix were actually more migrational in nature. So another, so back a few years ago in partnership with Axos space company, we're actually able to sort of do a little kind of LEO basic low Earth orbit experimentation by sending some of these bioprinted breast cancer models into like short flight, and then we were able to retrieve the samples after the flight, showing the viability of the cells. So it's pretty preliminary kind of experimentation, but opens up the possibilities in the future to allow space-based experimentation on these breast cancer models to allow investigations of how microgravity or other like space conditions like radiation are affecting breast cancer cell invasion and potentially therapeutic effects. All right, so then coming back to the microchannels, looks like it's a little. All right, so coming back to the microchannels, so we have been talking about these microchannels based on hydrogels, and they have been very instrumental in a lot of the application. But then there are two potential issues. One is that, I mean, these hydrogel kind of matrices can be expensive depending on what hydrogel that you use. But then second potential problem is that, I mean, once you have made these hydrogels, you have to basically use them pretty much right away. They cannot really be stored because of their degradation profiles and all the other kind of non-storable kind of parameters. So we have been thinking about how can you actually combine or transition these hydrogel-based hollow kind of tissue constructs to something other material that's more cost-effective and can be also stored on the shelf for a longer time. So then, I mean, paper-based devices then came into our mind very naturally because again they are very cheap materials, and then in the dry state it can be stored on the shelf for pretty much as long as you want, right? So then, with that in mind, we have been also working on some of the other kind of technologies based on the sacrificial bioprinting, but more embedded sacrificial bioprinting to allow creation of microchannels into paper-based devices. So here what happens is that you have these sacrificial bioinks based on petroleum jelly and liquid paraffin, and if you have them in the right ratio, you can actually directly print them into a matrix of bacteria cellulose, right? Basically, with the optimized conditions, you can basically print any type of structures, two-dimensional, three-dimensional, into this matrix of bacteria cellulose. So what happens then after is you just air dry these matrices to allow formation of a paper-like device. But not only is paper-like device, it's also having these embedded printed sacrificial templates, right? So because these are paraffin-based, you can actually increase temperature to allow their melting and then allow selective removal of this microchannel. I mean, this paraffin kind of templates from paper device. So now you can create microchannels that are perfusible, right? You can actually do multiple layers and also all different configurations as you wish. So then, as mentioned, I mean, these devices are pretty cost-effective, right? So each device at this dimension is only costing less than $4 US. So that's pretty cheap. And then, as mentioned, you can basically, in the dry state, store these devices on the shelf as long as you want until you want to use them. So when you want to use them, what happens is that you can basically autoclave these devices. So two things happen. So one thing is that you can rehydrate the device into a like hydrogel-like matrix that's allowing then cell seeding, but then at the same time you can also sterilize these devices with autoclave. So then with the reconstructed device or rehydrated device, you can then seed, for example, endothelial cells into the hollow channel configuration, and then in the surrounding matrix you can actually seed these breast cancer cells, creating a like vascularized breast cancer tissue at the bedside, right? Because again, you can imagine that you can use these devices on bedside, and if you directly get patient cells, breast cancer cells, or the other cells, you can rapidly create like we call point of care organ chip devices with a setup. Then start to really look at how the drug molecules are affecting the cancer cells in a time and dose dependent manner, potentially allowing bedside patient or personalized therapeutic screening for this application. So then, but another thing that we have observed in this first version of the paper devices that we made is that, I mean, the matrices are pretty dense, right? So these are basically bacterial cellulose microfibers that are packed together. So then, if you look at how the cells are behaving, these are breast cancer cells, right? So if you see breast cancer cells onto these matrices, I mean, the breast cancer cells are growing pretty well, but then they are only basically staying at the surfaces, not really going into the matrix. So it's more or less like a pseudo three-dimensional device, but not really truly three-dimensional device. So then with that, can you actually make this truly three-dimensional by doing something over there, right? So with that, can you actually do a bubbling process, basically foaming process to allow expansion of these matrices into 3 dimensions. So very simple process. What happens is once you have these devices in there, you can actually soak them into a solution of sodium borohydrate, and then that's going to create all the bubbles that are expanding then the layers of the matrix to a truly 3-dimensional structure that's highly porous. That's allowing then interactions of these different cell types in true 3 dimensions. So here's some examples. Basically, by controlling concentration of the sodium borohydrate, chemical or time that these devices are expanded, you can actually have thicker and thicker kind of expanded structures obtained at the end, right? So without expansion, the devices are usually in the range of a couple hundred micrometers, and after expansion can go all the way to a few millimeters in thickness, and you can see all the highly porous structure after expansion of the devices. So then with expanded devices, you can basically effectively increase diffusion profiles or capture effects of the liquids, not only from outside inward, but also from inward outside, right? So that's also allowing potentially better seeding of these cells after you kind of restore the devices. So with this configuration, I mean, we had to change a little bit of the bio-ink configuration. Again, I mean, even that, we're not seeing increase in the cost, right? So each device is still only costing less than 4 cents, which is pretty cost effective. So then if you do comparison with unexpanded paper device, if you see the endothelial cells into the microchannel, endothelial cells were pretty much confined within the matrix because there's no micropores in the surrounding kind of paper kind of microfibrils. But then if you expand the devices, then if you see the same endothelial cells in there, you can see very nice sprouting of the endothelial cells into surrounding matrix, forming again this multi-scale vasculature that's otherwise quite difficult to obtain with the device that's unexpanded. So now you can start to also again go back to do this vascularized by seeding breast cancer cells in the surrounding matrix, right? So now with expansion, you can see much better interactions of the endothelial cells and surrounding breast cancer cells in the true 3-dimensional space than the device with only without expansion, because you have all the highly packed microfibrils that are filling all the space in between these two cell types. All right, so then again, you can start to do the same sort of drug testing with this expanded sort of breast cancer model. All right, so then in the next maybe 10-15 minutes, I'm going to switch the gear a little bit now to talk about, still extrusion bioprinting, but more going into coaxial extrusion bioprinting of hollow or standalone hollow perfusible kind of tissue types. So so far I've been talking about sacrificial bioprinting. It's creating the hollow channels in there, but these hollow channels are tissue embedded because you have the matrix, always have the matrix surrounding these microchannels. So how can you actually make these standalone perfusible hollow tissues that's becoming something that's also very interesting to us? So here we designed a multi-layered coaxial kind of print head, which allows co-extrusion of the different layers of the liquids for creation of the hollow structure in the same step, right? So in this case, we basically delivered bioink in the middle layer and cross-linking agent from both interior and exterior layers, and then from there you can basically create a hollow, perfusible, standalone kind of tubular structure within a single printing process. And again, the biomaterials are pretty cytocompatible, so you can have cells in there. So eventually, with culture, you can form a very nice biologically relevant sort of hollow tissue structure with this bioprinting method. So here's some examples showing the process. So basically, by tuning the different diameters of the different nozzle layers and the different flow rates of different layers, you can also very nicely tune the diameter of the tubes or the wall thicknesses of the tubes. And with bioprinting, again, there's a robotic moving process. You can start to pattern these hollow tubes in both 2 dimensions and 3 dimensions, right? Basically, hollow fibers of different configurations. And as I mentioned, the biomaterials are pretty cytocompatible, so you can actually have cells in there. So these are actually vascular cells embedded into the surrounding kind of a wall kind of a matrix. And after I think 21 days of culture, you can see pretty nice biologically relevant formation of this vascularized or vascular bed that's being printed with this hollow coaxial bioprinting method. So then, as mentioned, it's a single hollow tube, right? So this is actually printed, I think, 10-layer tube structure, and the whole structure is actually perfusible because, again, you have a single tube that's entirely kind of connected. And then by further tuning the bioprinting parameters, you can build in also different variations in there. For example, in this case, we build in the tortuosity or curvature into the hollow tube structures, right? So basically, if you mismatch the bioink delivery rate versus the nozzle moving speed, you can start to pack these fibers more and more and create more and more curvatures into these fiber structures, right? So basically allowing you to model some of the vascular diseases or some of the like other more convoluted hollow tissues in human system. So believe it or not, I mean, these convoluted or curvy kind of hollow fiber structures also perfusible, right? Again, it's hollow inside and the entire structure multi-layered is actually also perfusible. So then another variation is more recently we have been working on a technique called shrinking bioprinting. Basically, if you directly print these hollow tubes, I mean, there's a limit you can reach, usually in the range of about a couple hundred micrometers in diameter. So you cannot really go below that in most cases. But if you print that tubular structure in the first place and then shrink the tube structure within a shrinking agent that we use, you can effectively make this tube structure much smaller in their diameters, right? So from a couple hundred micrometers, you can actually shrink these tubes all the way to about 30-40 micrometers in their inner kind of diameter, which is then becoming very physiologically relevant for some of the tissues that are of interest to model. Another variation is you can start to model not only the perfusible blood vessels, but also the single-end blinded lymphatic vessels that are allowing draining of liquid, right? So again, very simple modification of the printing method or the coaxial printing method. With that, you can then start to really seal one end of the tube that you print, and then create a lymphatic vessel-like structure that's produced with this hollow kind of coaxial bioprinting method, right? So these are on the top, these are basically entirely kind of perfusible tube structure that's mimicking the blood vessel. And at the bottom, you can see that we can create these single-end blinded lymphatic vessels of different sizes to model these lymphatic vessels to allow draining of liquid from the microenvironment. So now if you create a disease model, or in this case, more, well, initially more double tube configuration. And if you compare them, there are significant differences in terms of the transport phenomenon of these different molecules in this microenvironment. If you compare a single channel configuration, basically a single perfusible blood vessel, versus two-channel configuration, which is the blood vessel combined with the draining lymphatic vessel on the other side, right? Because again, you are really changing the microcirculation with this microenvironment. So what can you do with that, right? So you can actually then embed these tubular structures within a matrix that's having, for example, breast cancer cells. And then not only can you see, of course, differences between two-dimensional and three-dimensional culture, as we have been talking about a lot today, but also what's more important is that you can also see a significant difference between one-channel culture and two-channel culture, because again, if you have the doxorubicin, for example, here as a drug molecule infused into the blood vessel channel, there's a major difference in terms of transport of doxorubicin within this microenvironment that's causing a lot of differences in terms of the drug efficacy on the breast cancer cells that are like spaced in between these two vasculature. All right, so again, indicating basically how important it is to really allow creation of the biomimetic microenvironment for disease modeling and drug screening. All right, so then another sort of configuration or variation of the hollow tube bioprinting is that, I mean, I've been talking about bioprinting of a hollow tubular structure, but then it's hollow inside that's allowing liquid perfusion, that's mimicking, for example, vasculature and all stuff like that. But sometimes you also want to use that configuration to allow better generation of a tissue mimetic microenvironment within the microfiber, because you can use the microfiber then as the support matrix. Then within this microfiber space, you can now start to lower down your hydrogel content to very low concentration to allow really low stiffness kind of gelation to allow modeling of some of the key kind of tissue like disease phenotypes that's otherwise not possible to model with conventional bioprinting methods. So here, for example, we actually still use alginate as the primary kind of bioink for the outer shell, but then within this alginate microfiber that's hollow, we actually introduced JAMA plus cells within this microenvironment. But then because now you have the support alginate shell, you can actually lower down this JAMA concentration to a pretty low concentration, all the way down to like 1%. Again, that's if you do 1% JAMA, if you direct print that, that's usually not possible to print. But then if you embed that into a surrounding kind of shell that's hollow, that's serving as mechanical support, you can now lower down this matrix to a very low content to model some of the like key kind of disease phenotypes. So here we showed that with all these different types of the JAMA content in there, you can effectively modulate how the breast cancer cells are basically responding to these different stiffnesses of the JAMA matrix. But then more interestingly, recently we have been working to create a hollow, I mean, not hollow, but this microfibrous microenvironment that's modeling the bone marrow, because as we know, the bone marrow microenvironment is actually very low stiffness, right? It's only in the range of a few kilopascals, and that has to be used created with a low concentration JAMA that's again otherwise impossible to directly print with conventional bioprinting method. So here what happened is that we actually created a bone marrow microenvironment by introducing JAMA, very low stiffness JAMA, into the core of the microfiber, and then in the surrounding microfiber or shell we actually had alginate, but then alginate plus these nanohydroxyapatite particles to model the kind of cortical bone structure, right? So then with that, you can create a core shell structure having the kind of bone-like matrix on the surrounding, and then the bone marrow-like very low stiffness matrix in the very center. And then with that, you can actually create a disease model of the bone, which is myeloma in this case. So if you combine myeloma cells with the JAMA matrix in the center, you can start to model this myeloma in a very nice like biomimetic manner, right? So you can do co-culture or single culture. Single culture means that you can you only have the myeloma cells, but then co-culture means that you can have myeloma cell culture together with stroma cells that are widely present in the bone marrow as well. So then with that, you can start to look at how cells are responding to this microenvironment. Again, we show that three-dimensional co-culture is much more biomimetic than monoculture or conventional two-dimensional static, two-dimensional monolayer culture. And then you can also start to do drug testing with this bioprinted model that's very biomimetic. So basically, if you do drug testing with BTZ, which is a common drug that's used for myeloma, you can see a time dose-dependent toxicity. But then you can see that with co-culture, or model culture, there's difference, and also with three-dimensional bioprinted culture with conventional two-dimensional culture, there's also difference, right? So showing that our three-dimensional co-culture model is actually more relevant to the in vivo configuration. But then another thing that you can see, you can actually do testing for multiple drugs together. So rationale for that is if you see this image on the left corner, with a conventional kind of myeloma microenvironment, what happened there is that you do have a aggregation phenomenon of the myeloma cells because of the effect of the interleukin-6 that's created by the surrounding small cells, right? So then that's causing basically aggregation of the myeloma cells, but then also reduce the therapeutic efficacy. So with the another drug molecule TOC infusing there, you can effectively inhibit the aggregation of the myeloma cells, then start to really improve the therapeutic efficacy of BTZ, right? So that phenomenon can be readily reproduced with this bioprinted myeloma model that we have produced with this coaxial bioprinting method. So then what's more important is we showed that with the bioprinting of this bone marrow-like microenvironment, you can effectively prolong the preservation of patient-derived primary myeloma cells in vitro. So if you have this patient-derived myeloma cells obtained from patients, if you do two-dimensional culture in the monolayer configuration, then the cells are pretty much dying away very rapidly, right? So by day 3, already half cells are dead. By day 5, pretty much all the cells are dying off. But if you have the same cells that are then incorporated in the bioprinted model that's very biomimetic in terms of the bone marrow-like microenvironment, you can now start to really prolong their lifespan. So by day 7, there were still a ton of live cells present within this model in 3 dimensions, and you can then start to do all the kind of drug testing that we have that I have talked about in the last slide. All right, so before I end, maybe just spend a very quick few minutes talking about another bioprinting method. So far I've been talking about extrusion bioprinting that's have been very sort of simple to operate, but then allows you to really create different types of disease models of the human. But then the issue with that is, I mean, with extrusion bioprinting, you typically cannot make very complex structures with bioprinting, right? So then we have been also working extensively with this layer by layer based digital processing based bioprinting to allow really configuration of very sophisticated internal architectures with bioprinted tissues, right? So you can also, for example, incorporate all this multi-material capacity into DLP-based bioprinting as well. And with that, you can print very sophisticated, for example, solid tissue structures or this hollow kind of tissue structures, depending on the application. So in terms of disease modeling, we have been working on a specific example of liver disease modeling. So here, we showed that you can use DLP-based bioprinting to pattern these liver lobule-like structures in a very efficient way. You can do monoculture, meaning that you can have the hepatocytes embedded into the matrix, having only this kind of cells in there, and the cells will actually maintain its viability pretty nicely over days of culture. But they can also do co-culture as well, right? So basically, not only having the hepatocytes in the matrix, but also the stellar cells that are co-embedded into the matrix with patterning. And what's interesting here that while we see that the hepatocytes were more or less remaining within the lobule structures, the stellar cells were more migrating into surrounding kind of sinusoidal structure that's very much similar to the in vivo configuration of the liver. So now, if you look at functional characterizations, right? So the cells were having pretty nice affecting staining results, and also they were expressing highly of these cytochromes that are indicating their functionality of this bioprinted lobule-like structure. So then we can further induce them for disease phenotypes. For example, if you culture this patterned lobule-like liver structures with fatty acids, you can see accumulation of these fatty acids in the liver cells, creating a steatosis model. And you can look at how the drug molecules can reduce the fatty acid accumulation with this, within these liver lobule structures in a time dose-dependent manner. And the step forward, you can then also induce fibrosis with incorporation of TGF-beta in the culture, right? So basically showing the expressions of the collagen type 1 and also expressions of these alpha SMA in the stellar cells. And again, you can look at how the different drug molecules like rapamycin or curcumin are reversing the fibrosis kind of fibrotic phenotype in a time dose-dependent manner. Okay, and from there, I'm going to end by thanking all the wonderful collaborators across the globe, and also our funding agencies, and of course, most importantly, all the wonderful lab members that we have had over the past years who have done pretty much everything that I have talked about. And lastly, I want to also mention that the biofabrication now has an open special issue around the topic, basically placing 3D bioprinting into the context of human disease modeling. That's still open for submission. So if anyone's interested in special issue, please do get in touch with Richard and the journal to discuss about possibilities and also the timelines for submission. I'm going to end there, and thanks very much.
Thank you, Shrike, very much for that comprehensive overview of this emerging application of 3D bioprinting. Yeah, it was great to see, a summary of, those research results that were recently published and indeed forthcoming. So we'll now move to the questions and answers section of the webinar, and we do have some questions in. So we have a question relating to bioinks. What are the general design criteria of bioinks?
Yeah, so basically for bioinks, I mean, they are really dependent on different bioprinting modalities that are used and also the target applications. For example, I mean, extrusion bioprinting, you usually want the bioinks to be shear thinning or having higher viscosity so that it can actually extrude the bioinks in a better way to maintain the shape fidelity of the resulting kind of constructs. But then for DLP-based method that I talked at the very end, the inks have to be usually liquid-like, so you can then start to really cross-link them layer by layer. And then, yeah, really, I mean, depending on the final applications, for example, specific tissues to be printed, then there's also a specific sort of criteria on designing bioinks, because you have to really match the properties of the resulting tissue types to allow maximal kind of viability and functionality. So I mean, they're really dependent on what kind of modality that you use and also what type of tissue that you want to produce with the bioink.
Thank you. And a further question, would it be possible to combine different bioprinting modalities together to enhance performance?
Yeah, so that's actually a pretty interesting kind of trend or emerging trend lately with bioprinting. So yeah, again, I mean, each bioprinting method has its own kind of advantages and disadvantages. So basically, extrusion, for example, I mean, it's an easy setup, but then it doesn't allow a very complex bioprinting kind of structures. But then DLP, for example, gets you complexity, but then their setups are usually complicated. And the coaxial printing allows you to do the hollow structure, but then there's some other limitations over there too. So yes, I mean, I know that people have been thinking about that for a while, and I know, for example, there have been some publications around the corner where people have been combining, for example, extrusion bioprinting with DLP-based bioprinting. So yes, that's something that people have been thinking about, and I'm pretty sure that there are growing trends around this corner where people are trying to combine different modalities more and more in the future.
Thank you. And then one other question, which I suppose may relate to bioinks again. Can you explain the mechanism of hydrogel formation?
Hydrogel formation of the bioinks. So I think, again, that goes back to the bioink designs a little bit. So basically, well, again, depending on the different printing method, for example, again, going to extrusion bioprinting, then most likely, I mean, you don't have to have a photo cross-linkable version of the bioink for that extrusion bioprinting, for example, but the bioink has to be usually shear thinning or, again, higher viscosity to allow shape maintenance. But you can do a lot of the other cross-linking kind of strategies for extrusion bioprinted bioinks. But then if you do DL, yeah, then it has to be photoactive bioink so that you can do photo cross-linking layer by layer. But if you go for a method like coaxial printing, then usually we have the alternate component in there because it's coaxial, so you have to have multiple layers coming together, and usually that's being cross-linked by calcium chloride. So then you have to have alternate component in there as one of the components of the bioink. Yeah, so again, depending on different configurations or specific methods of bioprinting, then the gelation of the hydrogel formation kind of methods for this bioinks can be quite different as well.
Thank you. So we have an audience member thanking you for your nice overview. Their question is, regarding your hydrogel shrinking work, can you shrink gels containing viable cells, or do these have to be seeded post-shrinking?
Yeah, so yeah, good question. So I haven't got time to really discuss about that work today, but yes, we can actually shrink the constructs in the presence of cells. So what happens there is that it's a complex and charge complexing process. So so we see that there's a cell type dependent toxicity of this shrinking agent, but if you properly engineer the shrinking steps in the right way, for example, we have shown in our original publication that if you do multiple shrinkage that we call successive shrinkage, basically means that if you shrink for a certain time and counter for some time and then shrink another time, which is going to be the same size afterwards as if you were doing a single shrinking for a longer time, but then you're seeing much better cell viability in the successive shrinking process because again you don't exert excessive stress to the cells for extended period of time, but then you're doing a shorter shrinkage and then counter and then shrinkage again. So yeah, so there's some tricks to allow that to happen in the presence of cells. So yeah, so that's but that's something, again, it's early stage. We're still working on some other kind of works that are coming out hopefully next year to allow better explanation of the shrinking process and also potentially better allowing better shrinking in the presence of cells.
Thank you, Shrike. So a further question, what are the lifetimes of engineered in vitro tissue models? How long can they remain viable?
Yeah, so another good question. So the longest time that we have been culturing these engineered tissue models is about a month-ish, and they were pretty viable and functional during that culture period. We haven't been culturing them for longer just because we didn't have the need for that, but I know some of our colleagues have been culturing them for months in vitro, and they were still functional. So it's possible to culture them for pretty long, as long as you have all the kind of microenvironmental parameters correct. Yeah, but again, that really depends on the final application as well, right? Sometimes you want to them for being cultured for longer periods, but sometimes you don't need them for that long. Yes, again, that's application dependent problem.
Thank you. And then as a further question, would it be possible to combine different bioprinting modalities together to enhance performances?
Yeah, I think we briefly discussed about that. So again, each printing method has its own unique advantages and disadvantages. So yeah, so it's been really a hot topic recently by the community where people are trying actually quite hard to combine different bioprinting modalities together to allow maximum performances. Yeah, but again, that's still beginning, but I hope that we're going to see much more advancements in the years to come.
Thank you, Shrike. We are almost at time, so I'll just move to one more question. I'm not sure if this has been answered previously. What is the size of vascularization currently available with bioprinting?
Yeah. So again, I think depends on the bioprinting methods, right? So if we do sacrificial bioprinting, we're usually looking at about a couple hundred micrometers of diameter, and that can go down to maybe tens of micrometers with some certain configurations. If we're talking about, for example, hollow this coaxial bioprinting, we're looking at usually also a couple hundred micrometers as well. If we're looking at the DLP-based bioprinting, DLP allows you to really create multi-scale vasculature, going from bigger vasculature to smaller kind of channels. But then I think they're still limited to a couple hundred micrometers. So that's why we have also, again, been developing this shrinking method to allow really shrinkage of the channels after bioprinting, so you don't have to really change any of the hardware configurations or software configurations of the bioprinters or the bioink designs. But then with our shrinking printing method, we showed that we can actually reach almost single capillary level kind of size in the range of like 10-ish micrometers. So that's possible too. But then again, really again goes back to the specific applications. Sometimes you want to have smaller vessels, but sometimes you don't want the vessels to be going at the small scale. But another possibility is that, as mentioned in my talk today, you can also allow spontaneous sprouting of this vasculature to create microvasculature. So you can have these microchannels that are endothelialized with the cells, and then allow them to sprout out in surrounding matrix, form this multi-cycle vasculature. So I mean, there are different methods that can be used, and that really depends on, again, the modalities to use and also the final application that are intended for.
Thank you. Oh, and we've just received another question briefly. Do you think that we'll be able to study the allergies and the environment via an organ on a chip?
Yeah, good question. So definitely. So again, that's why we have been extensively working on combinations of bioprinting and this microchip devices, which I didn't get time to present today. So again, bioprinting, for example, gives you a lot of the chance to really recapitulate the three-dimensional microenvironment. But then in terms of incorporating the dynamic cues, bioprinting is not as effective as the microfluidic devices. So on the other side, I mean, microfluidic devices can give you the really chance to incorporate all these dynamic cues like the fluid flows and also the mechanical actuations, but then they are not as good as bioprinting to allow three-dimensional configurations. So that's why we have been really interested in combining different fabrication methods to allow really in vitro recapitulation of this human tissue and organs and also the disease types to really allow biological studies and also drug screening applications. So yes, I mean, if with all those kind of things come together, you can meticulously design how these tissues are in terms of configuration, function, the viability, and other things like that, and start to do not only drug screening, but also, of course, all the fundamental biological studies as well with those.
Okay, thank you very much, Xiao, for responding to the audience questions. So in the interest of time, we'll now move to close the webinar. So I'd like to thank the speaker for giving his talk today. And I would also like to thank the audience as well for participating and putting forward your questions. So thank you all.
Thank you.
Thank you.